Hello everyone, welcome to the channel Microbial World. So in this video we will be discussing dark field microscope. So the dark field microscope, it is used for the observation of living unstained cells and microorganisms. So in bright field microscope we have told that in order to increase the contrast of the image we need to stain the specimen, right? But in dark field microscope, using this microscope we can observe unstained samples itself. Okay, and how does the image appear in a dark field microscope? Here the specimen will be brightly illuminated and the background will be dark. So this is just opposite to that of the bright field microscope. What we said in bright field microscope, there the background will be bright whereas the specimen will appear dark. But here it is the exact opposite. Here the specimen will be brightly illuminated, okay, whereas the background will appear dark. And another important thing regarding the dark field microscope is that it is using a special type of condenser that will scatter the light so that the light will reflect from the specimen at an angle. We will say more about this in the principle. It is using a special type of condenser. So these are the main things regarding a dark field microscope. So now let's see the principle of dark field microscope. So you can see there, here is the light source, condenser lens, the specimen, it is placed on the stage of the microscope, the objective lens and the eyepiece. Okay, so the light source, it is producing light. And we have said that the dark field microscope is using a special type of condenser. That is because just beneath this condenser lens there is an opaque disc, okay, opaque disc, or it is also called dark field stop. It looks somewhat like this. So this opaque disc, it is blocking most of the light that is entering this condenser lens. It will block most of the light and will produce a hollow cone of light. You can see here. It is producing a hollow cone of light. Now this dark field microscope, it is designed in such a way that the light does not directly enter the objective lens. Okay, so see, the dark field stop or this opaque disc will block most of the light and will produce a hollow cone of light, and that light, it will hit the specimen at different angles. And the main thing is that only the light that is reflected or scattered from the specimen, you can see here, only the light which is reflected or scattered from the specimen will enter the objective lens, then goes to the eyepiece and we can observe it. The light that is not hitting the sample, that is, see, this unreflected light, it will just pass around the objective lens. Okay, it will pass around the objective lens. So this is the main thing that you need to understand. Only the light which is scattered or reflected from the specimen will enter the objective lens, and the unreflected light, it will just pass around the objective lens. So how does the image appear? The specimen, it will be brightly illuminated and the background will be dark. Okay. So the advantages and disadvantages of dark field microscope. The advantage is that the resolution is so much better when compared to that of a bright field microscope. Okay, improved image contrast. So even without staining the specimen, we can improve the image contrast. It also helps in detecting non-culturable bacteria that is present in the patient samples. And no sample preparation is required, no special setup is required. Okay, and the disadvantage is that the specimen or that sample has to be strongly illuminated. This can damage that specimen, and the low light level that is seen in the final image is a disadvantage. And also, if any kind of dust particles is present, these dust particles can also scatter light. This is also a disadvantage. Uses of dark field microscope. So the dark field microscope, it is used to visualize spirochetes. Spirochetes such as Treponema pallidum, which causes syphilis, Leptospira interrogans, which causes leptospirosis. So these spirochetes can be visualized using this microscope, and also we can observe the motility of the microorganisms. So using this microscope, we can observe flagella in unstained cells. Thus, we can observe the motility. It is also used to observe internal structure of larger eukaryotic microorganisms like algae, yeast and all. This microscope, it is also used to study marine organisms such as plankton, diatoms and algae. It is also useful for the demonstration of very thin bacteria that is not visible by ordinary illumination. We can demonstrate very thin bacteria using these microscopes. So these are the main uses of a dark field microscope. So we have discussed about the principle by which a dark field microscope works, its advantages, disadvantages and uses of the dark field microscope. Thank you.